Bio 150 Lab students. This is Dr. Cass, and today we're going to be talking about PCR. In particular, we're going to be talking about the role of primers in PCR. This is going to set you up for the activity that you're going to complete during the second half of your Week 6 Bio 150 laboratory. Hopefully, it will also help you to have a better understanding of the mechanistic basis of the PCR reaction. Up until this point, we've been talking about why PCRs fail. The truth is, PCRs can fail because of any of the components of your reaction mixture. They could fail because of issues with what we call our reactants, such as a missing component, or a component that's gone bad or is at the wrong concentration. We control for problems in our reactants by doing positive and negative controls. Our positive control reaction tells us whether our reaction mixture was assembled correctly and all of the components were still good. If you get a band in your positive control lane on your gel, that should tell you that your reactants were just fine. Last week, we talked about problems with your template DNA or your DNA extract. We said that problems with the template come in two major flavors. Problems with the DNA concentration, either too high or too low, and problems with the DNA purity, where you might have PCR inhibitors present in your DNA extract. Our best solution here was to use the nanodrop spectrophotometer to assess both the concentration and purity of our samples. The next step of that solution was if we recognized that our concentration was too high or our purity was too low, we could dilute our DNA extracts to increase the chance of our PCR working. If our DNA concentration was too low, then perhaps we would choose to re-extract our DNA and attempt to get a higher DNA concentration. The component that we haven't yet talked about are the primers. The primers are an incredibly important part of a successful PCR reaction, and they're probably the component that you've thought the least about up until this point. Problems with primers also come in kind of two different flavors. One is that the primer sequence matches your template in too many places in the genome. And the second is that your primer sequence doesn't match the template anywhere in the genome. Both of these things can cause major problems for your PCR. So how do we solve them? We solve them by confirming that our primers match the genome of our organism of interest in the appropriate region of the genome, and that they only match in a single location in the genome. Now, in order for us to talk about how we're going to do this confirmation, we need to back up, and we need to talk about the structure of DNA and the specific way that primers actually work in a PCR reaction. So you'll recall that DNA is usually found as a double-stranded molecule. Often, you're only given a single DNA sequence, one string of A's, T's, C's, and G's, but this is actually representative of a full double-stranded DNA molecule. The reason why you are often only given a single sequence as a representation of this more complex structure is that you can predict the second strand of this double-stranded molecule from only a single sequence. In order to do this, you need to know two rules. One is that double-stranded DNA molecules are formed of complementary base pairs. Guanine nucleotides pair with cytosine nucleotides, and adenine nucleotides pair with thymine nucleotides. So anywhere that you have an A in one strand you will have a T in the other strand. Anywhere that you have a G in one strand, you will have a C in the second strand. But this isn't enough information for you to make a full prediction of the second strand. You also need to know something about these designations on the ends, 5' and 3'. What these indicate is that each of these strands has what we call directionality. One end of a DNA strand is not chemically identical to the other end. This has to do with the structure of the sugar phosphate backbone of the DNA molecule. At one end of a DNA strand, you have a free phosphate group. This is referred to as the 5' end. At the other end, you have a free hydroxyl or OH group. This is referred to as the 3' end. When you assemble a double-stranded DNA molecule, the two strands have to be in what we call anti-parallel arrangements. You can think of this as kind of opposing or opposite arrangements. When one strand is drawn with the 5' at the left, the second strand must have the 3' at the left. This is the anti-parallel arrangement of two strands of DNA. This is important when you're predicting the second strand because you need to make sure to designate which end you are starting with when you start to write the sequence. If you were to move this 5' over here and start with TAC and say that this strand was going to be complementary to this one, your answer would be incorrect. Strands are only going to bind together to form a double-stranded molecule when the complementary base pairing is in an anti-parallel arrangement. Here's a DNA sequence for you to think about for Canvas quiz question 1. Open the Canvas quiz that's associated with this video and go to question 1. It asks you to predict the second strand of the DNA molecule given this first strand. Pause the video and think about the answer. Once you think you have the answer, then come back and we'll talk through why one answer is correct. Okay, the correct answer to Canvas quiz question 1 is 3' CACAA. Remember, we have to have complementary base pairing when the strands are anti-parallel. Up here, we started with 5', so now we're starting with 3'. This sequence could also be written as 5' ATTA, going left to right as well. That would also be considered correct. Now that we've reviewed DNA structure, let's talk about the role of this DNA structure in a PCR reaction. Now remember, PCR generates many copies of a particular region of the genome. And it does so by repeated cycling through 3 steps: denaturing, annealing, and extension. During the denaturing step, the double-stranded DNA template is heated to 95 degrees Celsius. Heating a double-stranded DNA molecule actually breaks the chemical bonds between the two strands and allows them to separate into single-stranded DNA templates. This is important because DNA polymerase, the enzyme that's capable of copying DNA, can only work on a single-stranded DNA template. After denaturing, the second step is annealing. Annealing is important here because this is where the primers will actually bind to our DNA template molecules. Another rule about DNA polymerase as an enzyme is that while it needs a single-stranded template, it actually can't start on a single-stranded template. It needs a short double-stranded region in order to start the synthesis reaction. This is what the primers do. We have 2 primers per PCR reaction. One of these primers will bind to each of our template molecules. When they bind, they have to follow the same rules as any double-stranded DNA molecule. They must be complementary in an anti-parallel arrangement. During annealing, we lower the temperature of the reaction to allow the primers to bind to the template molecules. Now that our primers have come in and bound and made a short double-stranded region, the DNA polymerase enzyme can latch onto this and begin copying the single-stranded region of the molecule. DNA polymerase always extends from the 5' end of the newly synthesized molecule to the 3' end of the newly synthesized molecule. So when extension occurs, it will occur in this direction for this template and in this direction for this template. Here we go. That's extension. During extension, the DNA polymerase molecule is adding new complementary nucleotides to this strand. When extension finishes, we now have 2 double-stranded DNA molecules again. But this time, they're kind of asymmetrical. Our newly synthesized strands are shorter than our original. And this is because the polymerase can only extend in a single direction. The newly synthesized strands are shorter on the 5' end, here and here. The double dashes are indicating that these DNA molecules actually terminate at this point, where the single dashes are indicating that the DNA sequence goes on off of the screen in that direction. Now, PCR cycles. You're used to hearing denature, anneal, extend, and that's about it. But the truth is that when you run a PCR reaction, it's actually denature, anneal, extend, denature, anneal, extend, denature, anneal, extend, something like 30 times. When you start the second cycle of a PCR, for every original double-stranded DNA template molecule that you started with, you now have 2. So instead of only having 2 single-stranded templates to start with, we now have 4, and they're not all the same. We have our original template molecules, which are very long, and then we have our 2 newly synthesized molecules, which are shorter on the 5' end. Let's focus in on those newly synthesized molecules to think about the way that primers define amplicon length. These templates are still here. Don't forget about them. We're just going to ignore them for the moment. So here's our newly synthesized template molecules going into PCR cycle 2. Now when the primers come in and anneal, they're going to anneal to the opposite end of our PCR amplicon. There and there. Now what's going to happen when extension occurs? Remember, we go from 5' to 3', and the double dashes indicate that this DNA molecule ends at this point. So now when extension occurs, the polymerase runs right off the end of the DNA template molecule. It can't go any further. Because it can't go any further, we now have an even shorter new DNA molecule that's been synthesized for both of our original templates. The length of these two newly synthesized molecules at the end of cycle 2 will be identical, and it will be equal to the length of the forward primer plus the length of the reverse primer plus the number of nucleotide bases in between their two binding sites. By the end of the second cycle of PCR, you have defined your amplicon length. Again, it's the length of the forward primer plus the length of the reverse primer plus the number of nucleotide bases in between their binding sites. In this example, the amplicon length would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 nucleotides long. You can see now that the position of the primer binding is incredibly important. It is the defining factor in determining the size of your PCR amplicon and the size of that band that you're going to get on your gel. Let's look at another example. So here's an example where we have 60 base pairs of the hypothetical genome of our critter of interest. And we're going to design primers against this particular organism's genome. We've chosen the regions that are shaded as the regions to design our primers against. One of these regions, the one towards the more 5' end of the sequence, will be our forward primer binding sequence, and the one towards the more 3' end will be our reverse primer binding sequence. Now remember from the example before that we had a double-stranded DNA template, and one primer bound to one of those strands, and the second primer bound to the second strand. When you're designing primer sequences, this is important because your forward primer is actually going to be identical to the sequence you're designing off of. The reason for this is because this sequence that we have here is already the reverse complement of the second template sequence, right? This guy is actually a part of a double-stranded DNA molecule. And if we want our primer to be complementary to that second strand, then our primer should be identical to this sequence. What about the reverse primer? The reverse primer we actually want to bind to this sequence that we've been given. If we designed our reverse primer as being identical, that wouldn't work. The reverse primer needs to be what we call the reverse complement. That is, if we start at 3', then we'd be starting at 5'. TTT would become AAA, and CCC would become GGG. This sequence is complementary in an anti-parallel arrangement to this proposed primer binding site. Now, when we run our PCR, we should be able to predict the size of the amplicon that we would get given our primer binding sites. We do this by adding the length of the forward primer, the length of the reverse primer, and the number of nucleotide bases in between those primer binding sequences. So, how big do we expect our amplicon to be given these two primers and this particular genome? This is Canvas question 2. pause your video for a second and come back when you think you have an answer. The answer here is that we would expect our amplicon size to be 60 nucleotides long, because we know that this entire sequence is 60 nucleotides, and we designed our primers all the way at the ends. If you counted up all of the nucleotides, including the length of the forward, the length of the reverse, and the nucleotides in between, you would find that it would be 60 nucleotides long. Now, when we actually run this PCR, we end up with a problem. Here's our marker lane with our 10, 20, 30, 40, 50, and 60 nucleotide markers. Here's our band that we expected at 60 base pairs long. But we also got a second band at 20 bases long. This is bad. If you end up with two different amplicon sizes, you can't sequence that reaction. Good primers are designed to bind to only a single location in the genome and produce many copies of only a single PCR product. These primers produce two different PCR products. Why might that happen? What's wrong with our primers? What's wrong with our primers is that these primers bind to the genome in multiple locations. If you look closely at this genomic sequence, you'll find the second primer binding site right there. So notice that our forward primer binds to our genome in two different locations. And if you add up the number of nucleotides in between the second binding site and the reverse, you'll see that you would anticipate a 20 base product. If you run into multiple binding sites for a primer in your genome of interest, the solution is usually to design a better primer. In this situation, if we simply scoot our proposed binding site down a little bit and design a new forward primer that binds to one and only one place in the genome, we will end up with a single PCR product of 56 nucleotides in length. By checking ahead of time whether your primers bind to multiple locations in the genome, you can avoid ending up with getting multiple bands on your gel and having to redo everything. What about the second problem that we can have with primers? We answered what can happen when the sequence matches the template in too many places, but why might the primer sequence not match the template at all? The reason for this is that nucleotides change over evolutionary time through the process of mutation. Organisms don't have identical genomes, and they're constantly accumulating new mutations. Our primers that we use in this course have been designed against specific regions of the genome that are highly conserved. That is, regions that don't accumulate mutations very quickly. But that doesn't mean that they don't accumulate mutations at all. They do. In this situation, we have a perfect match between our primer and our template, where every nucleotide is perfectly complementary. But as species diverge over evolutionary time, mutations can accumulate, and our primers might not match as perfectly as we would expect. So for instance, here's a template taken from a different species. In this situation, here's our primer, and we can see that there are specific regions that are no longer perfectly complementary. Primers can work in situations where they are not a perfect match for the template, but they start to work less well. And if you accumulate too many mutations in the primer binding region, the primer may not match at all, and it may not be able to amplify the genome of a particular organism. By checking our primer sequences against the genome of our organisms of interest, we can make predictions about whether our primers are actually going to be able to successfully amplify DNA from that organism. Here's Canvas quiz question 3. Both plants and vertebrate animals have a cytochrome C oxidase subunit 1 gene, the gene that we call CO1 in DNA barcoding. However, the primers that we've designed for vertebrate animal CO1 will not amplify even the purest of plant CO1 DNA. Which of the following statements best explains why these vertebrate animal CO1 primers will not amplify the plant CO1 gene? Okay, we're going to give you the answer, so pause it if you haven't finished. The answer here is that plants and animals are very, very distantly related. They've been diverged for over 1 billion years, and the primer binding sequences have also diverged over evolutionary time. New mutations have accumulated in the plant and animal lineages in the primer binding region, so that our vertebrate animal primers are not capable of binding to the plant CO1 DNA. Okay, so we've talked about all of these reasons why our PCRs can fail. And we've done our positive and negative controls, and we've nano-dropped our samples to check for our template. Now we're going to do an activity where we actually check our primers against the genome of our organisms of interest. This will allow us to predict whether our primers will bind successfully to one and only one region of the genome, and it will also allow us to make a very specific prediction of the size of our PCR amplicon. This is activity 4 in your week 6 handout. For activity 4, you're going to complete this assignment as a part of your laboratory notebook. Your instructor will hold a synchronous Zoom session for the second half of the week 6 lab, after the face-to-face students have set up their PCR reactions. Attendance at this Zoom session is strongly encouraged. This activity can be quite challenging. When you complete the activity, you can work together as a group, and we encourage you to do so. But every student needs to download the APE software and work through the activity on your own. Multiple students should not submit the same screenshot for the laboratory notebook entry. Here's your last Canvas quiz question about the primer mapping activity. The answer to this is false. Everybody in your group needs to work through the primer mapping activity and submit a unique screenshot to the lab notebook assignment. We want to make sure that you all get an opportunity to work through this because it will improve your ability to understand the role of primers and how they can allow you to predict your amplicon size. Good luck with your PCRs. Good luck with your primer mapping activity, and I hope you're enjoying your Bio 150 lab.